Okay, welcome everybody to our session. And it looks like we can see chat boxes, so thank you. We are ready to go here. And I want to welcome you into this session with Vanessa Light and Jason Button from Illumina. The future is bright, bringing genomics and DNA into your classroom. I'm going to begin by introducing our speakers. We work very closely here at Biocom Institute with Illumina and Vanessa Light here. She's the program manager on the corporate social responsibility team at Illumina. Focusing on STEM education programming and outreach, Vanessa has helped to create and grow the outreach programs for both nonprofit partners and Illumina employees. And Illumina is committed to equitable access to STEM education for all, with the goal of empowering students to envision themselves as future scientists, innovators, and trailblazers. Thank you and welcome, Vanessa. And Jason Button is a senior sales enablement content specialist at Illumina and the host of Illumina's Genomics 101 at Home. As a genomics professional who is passionate about access to genomics education, Jason has hosted Illumina's Genomics 101 at Home, visited classrooms virtually leading strawberry DNA extractions, and helped launch Illumina's inaugural The Future is Bright campaign. So I will let them begin. Thank you guys for being here, and we will get started here.
Thanks, Marissa.
Perfect. Thanks, Marissa. So let me see if technology is on our side, and I will share my screen. So as Marissa said, my name is Vanessa Light, and I have the pleasure of being on the Illumina Corporate Social Responsibility team, something I'm proud of and absolutely love doing and being able to connect with all of you. So, you know, a couple housekeeping items that Marissa had mentioned, but it will be recorded, and the session will be recorded and available if you want or need to reference it at a later date. And then we also have that Q&A box. So think of me as kind of your program manager and Jason as your science head. So we'll be able to answer questions throughout the presentation and then also at the end. So the more questions, the better. So don't be afraid to ask them. We'll save all the hard science questions for Jason. And I just want to make sure, can everyone see my screen?
Not yet.
Okay.
Yeah, and please feel free to put questions into the Q&A box. We will take some time and space throughout to answer those. So we'll be taking a look at those.
I think we're good now. Yep.
Yep, we're good now.
Okay. So let's start with our agenda and what, sorry, too many windows open. Okay, there we go. So really we're going to be sharing a little bit about Illumina, our mission, why connecting with you all is so important to us. And then Jason's going to dive into some of the Illumina breakthrough technology that makes us so unique, and also just in the sequencing technology. So he'll be talking about the technology, how our customers use it, and then we'll be sharing how you can use ready-to-go resources in your classrooms to share about DNA and genomics. So let's get started. We have Marissa that's going to be putting up a few polling questions. So we're going to do our first one. And on a scale of 1 to 5, 1 being a newbie and 5 being advanced, what is your level of comfort teaching about genomics?
So we just popped that in the chat box. So please feel free to put your score in the chat box. We have a 4, a 4, a 4, a 1, a 3.
And we realize that we have some educators, we have some industry, individuals, we have a whole gamut of individuals. So there's no wrong answer.
We've got a 3, a 2, another 3, another 4.
Perfect. So it looks like we have a good mix, which is great because we have options for everyone. So for different age groups, different entry points, different mediums, you know, some of us are in a virtual setting, some of us are in person. So we'll be able to share lots of different options. So one of Illumina's main philanthropic focus areas is STEM education. And by partnering with educators like you, we can really work to achieve our goal of inspiring the next generation of those scientists, those engineers, and the innovators to pursue careers both in life science, in those scientific and non-scientific careers. And so our hope is to support teachers in feeling able and ready to bring genomics into their classrooms, which leads me to our next polling question, Marissa, if you can put that in there. What are some of the barriers that prevent you from bringing genomics into your classroom? And again, there's no wrong answer. We just, we want to hear from you and we want to be able to connect with you.
Supplies, money, and time.
Was that 3 people or 1 person?
1 person. Cost required to cover content. Supplies, time, short on time, having to get through so much other material, time, hands-on experiences are costly, and content knowledge. And then computing resources, development of curriculum. That's all our answers. Oh, 1 more. Hard to run labs with classes of 30 to 40 students.
Hard to do anything with classes of 30 to 40 students. So it seems pretty consistent with what, you know, some of the things that we've heard in the past, and then also obviously what we're hearing today. So the first that we hear is that it's a fast moving industry and that it's hard to keep up with the latest technology. And I think that really ties to the cost. You know, keeping up with the cost, keeping up with the knowledge, keeping up with curriculum, you know, it's not the only thing you are all teaching. So the second one is, you know, it's a newer technology. So educators did not cover applications of genomics in their higher education. And then the third that we hear a lot is that educators do not feel confident in the content and cover foundational topics like DNA, chromosomes, and things like that. So they cover those, but they don't dive into the applications, the how and the why. So we're going to let Jason, we're going to hopefully cover some of those challenges with some programs that we have later in the session. But to set that scientific foundation, I'm going to hand it over to Jason to talk a little bit about Illumina, our technology, and genomics. So I will start a video and he can talk through it.
Hey everyone, yeah, so this might be a little bit of a refresher, but DNA can be found really in any living thing. So this is the human body, and we're going to kind of travel into the body and see where we can find it. So as you travel into a blood vessel, in this case, you're going to see a bunch of cells floating around the blood vessel, and really any cell that contains a nucleus in the body will contain DNA. And so as we enter this white blood cell, we're going to see a bunch of chromosomes that are floating around the nucleus. And DNA is essentially just tightly wound strands of DNA, or chromosomes are just tightly wound strands of DNA, and they're going to be double-stranded. So as you unravel that, you start to see the makeup of them. And all DNA is made up of A's, C's, T's, and G's that combine to form this double helix that gets wound up. And so any living thing is going to contain DNA, and you can extract that and sequence it to see what that organism is built for, and how that organism, like what sort of features it might have or traits it might have. So if we go on to the next slide, Illumina sequencers, what we are doing is actually reading that DNA. So all the A's, C's, T's, and G's, and using that to infer all sorts of things. And the great thing about that is that the applications are kind of countless. Like there really is no limit to what we can do with it, especially given today's analytics and computing powers, because it's a lot of data. And so, but each of the applications kind of fall under 3 main categories. I'd say most of them do. So the first one is whole genome sequencing. And this one's kind of easy to understand. When you're talking about a genome, it's all DNA that's found in one organism. So if we were talking about your personal genome, we're talking about reading your full genome. And applications that are useful for with whole genome sequencing are resequencing, which is basically taking one full genome from a patient and then comparing it to a reference genome and pointing out maybe some mutations that could be causing diseases in that patient. So think rare undiagnosed genetic disorders. So maybe a child growing up is having some symptoms and we're not sure what's causing them. We can sequence the full DNA and point out where some mutations are that could be causing these symptoms. There's also de novo sequencing, which can really be easy to understand as well. It's just sequencing a new genome. So at some point, there's going to be a time when an animal or an organism or a plant is going to be sequenced for the first time, and that's called de novo sequencing. So you're not comparing that genome to a reference genome that you've made before. You're actually building a full genome out of what you've sequenced. It's kind of like a puzzle. You're fitting the pieces together. And then the next type of sequencing is targeted sequencing. So a whole genome, you're sequencing the whole thing. Targeted sequencing, you're actually pulling out portions of the genome that you want to take a look at. And why this can be useful is that, say if we know that there is a list of genes particular to cancer, so mutations in these genes could be cause an increase of a chance of cancer in a patient, then we can actually pull out those genes, sequence them, and look for the specific mutations. This makes diagnosing quicker because we're not looking at all the data in the human body, and it is also cheaper because we're actually doing less work essentially. We can also do things like exome sequencing. So whole genome sequencing is sequencing the whole genome again, but exome sequencing is when scientists have kind of determined what is the coding region of the genome, which means what parts of the DNA actually become genes and actually are turned into proteins in the human body typically. That's going to be your exome. So by just sequencing that, we can kind of get rid of all the junk that's sort of some extra letters left behind due to evolution and really get into what might be impacting your human body. And then the final one is RNA sequencing, which is really cool as well. And so what this does is that the central dogma of biology and DNA is that DNA is transcribed into RNA, and then RNA is translated into proteins, because essentially what you're trying to do is get from DNA to the proteins that make up your body, and the middle man is RNA. And so by sequencing RNA, you actually get a snapshot of what's being made in the body at a certain time. So say if we're just sequencing RNA, we can get an idea of what proteins are being upregulated and what the body is actually producing, which can go into things like cancer research and rare undiagnosed diseases as well, to see what sort of proteins the body is actually making and if there's any sort of issues there that we might need to look into.
We do have a question.
Yeah.
If you sequence a person's whole genome, would you also ever do targeted exome and or RNA sequencing as well?
Yeah, so that's a really good question. And typically when you're thinking about a diagnostic journey is what we call them when we're talking about actually going from a patient with no genetic sequencing to trying to figure out what their diagnosis is. A lot of times they'll start with that targeted sequencing just because it's cheaper and quicker to get those results. But then you'd move on to whole genome sequencing. Say if you weren't able to find what the cause of these symptoms were and you realize that you need to look at the whole genome. There wouldn't necessarily be a reason to go back and do targeted sequencing unless you needed to find like a really rare variant or one that wasn't as present in the body and do what's called deep sequencing, which is reading the same region over and over and over again to really look into that and get a lot of data. But if you do whole genome sequencing, typically you'd be able to find your answers if the analytics are out there. I hope that answered it. All right, and we can go on to the next slide. So when you're talking about the journey of one sample to an application, it starts in the lab. And so it always starts with scientists extracting DNA from a sample. And this could be a sample of a blood draw, a tumor sample. It could be samples from plants or crops or livestock, anything that's living. And so scientists will extract that DNA, do what's called a library prep to prepare that, to prepare it to be read or sequenced on one of the Illumina sequencers, and then loaded onto our instrument. And what we can infer from this, if we hit enter, is a lot of different things. And so doctors, we're kind of on the precipice of precision medicine because of this. So doctors can start looking at your genetic makeup to determine what exactly is causing your disease. And beyond that, they can also take your genetic makeup and decide, all right, does medicine A make more sense, or does medicine B make more sense based on your genetics? So would a specific type of cancer respond to a medicine? If the answer is no, why even go down that path? We can say medicine B is going to drive results based on past clinical studies. And then we can also go into ancestry. So we can, you've probably heard of 23andMe and ancestry.com. And so we can start to determine where families are coming from. There's families that are reunited because of genetic testing. And not only that, but there's also non-invasive prenatal testing, which is a really exciting up and coming application where essentially there's enough of the baby's DNA that's floating around in the mother's blood that you can actually test the mother's blood with a blood draw, sequence the baby's genome, and determine if there's anything that should be concerned, that the parents should be concerned about before the birth. And this type of stuff, it sounds really like it's going to come along in the future, but it's happening in doctors' offices today around the world, which is incredibly exciting, all from a blood draw. And then it's not limited to human health or human bodies just in general. We're also talking about making sure that the world's agricultural crops are resistant to pests based on certain traits that plants might already exist in nature. We're talking about making sure that our livestock stay healthy in changing climates. And so we're really making sure that we can actually feed the world with our current technology. And so you might not, some of these concepts and some of these applications might sound new to you. And that's really because in the past 20 years or so, the cost of sequencing has gone down incredibly quickly. And so when we first sequenced the human genome back in 2003, the project took 11 years, and it was a $3 billion effort in order for scientists around the world to make all this data and collect it all and combine it all for one genome. Huge effort. Not something that people were going to be doing individually. And so we came around, we started making some sequencers and trying to drive some of the costs down. And so you'd see some other technology bring down the cost to like 20 million and then down to $2 million. And then we came out with our genome analyzer in 2008, one of our first commercially available DNA sequencers. It brought the genome down to $200,000, and it says the first 30x genome because that's the standard of genomes today, where you're not just reading the genome once, you're actually reading it 30 times, and so that you can confidently say each base is read accurately because you've actually read it 30 times. And then in 2010, we came out with another instrument so that it's now $10,000 per genome. Then in 2014, we brought the price down to $1,000. And then in 2020, it's down to $600. This is really getting to the point where even individuals who might suspect that they have a genetic disorder, if it's really impacting their life, they might be able to fund their own genome sequence. So they could actually see what it is and then go and find doctors who would be able to do it. But it also opens up the doors for companies to start, for academic labs to research more about the genome. And so that's why we're seeing this explosion of applications come out, just because of the cost has actually been driven down significantly. And then next slide. And so when you're talking to your students, or if your students hear about the word genomics or genetics, they might first think of like a biologist or a chemist, which are really kind of low-hanging fruit scientific careers that they can go for. But what I want to show with this slide is that it doesn't take just biologists and just chemists or scientists to create a company as large as Illumina. We really need specialists, so microbiologists and immunologists, which you may not think of right off the bat as people that Illumina would hire. And then we have a slew of engineers, optical engineers, because our systems actually work by imaging DNA. We need software engineers to run the instruments. And then you go into making the product. So you think of your manufacturing engineers and your manufacturing technicians, but did you think that we had technical writers who have to write the whole manual on how to run the instrument, or a materials manager to determine if we have enough material to actually make or manufacture the product? And then we also have delivering the product. So we have lab services, which we have lab assistants for, but we also have sales reps who a lot of them have PhDs. We have marketing program managers who need to understand the instrument in order to make content for sales to actually sell the instrument. And then I'm in the sales enablement group, and I take that marketing content, make sure that it's fed easily to sales teams, and that we can actually sell our instruments. But then there's also a lot of non-science background jobs. So think of a customer service rep who is making sure that orders are delivered properly, and then public relations specialists. And then supporting customers, this is actually where I started. I started in tech support. It was not a job that I was thinking that I would do when I was in high school, but I loved it and it was fantastic. I got to chat on phones, but I really needed to know a lot about the product, and I really needed to know a lot about the science, because I was talking with scientists around the world on how to run our instruments and troubleshoot any issues. And then we also have web developers and web designers, and then a whole slew of others who are supporting the company, who each have their own individual, maybe not so sciencey background, but they each know what they're doing, and it makes Illumina run like a well-oiled engine.
Amazing, Jason. It's amazing to hear you talk about that in just such a concise manner, but it makes sense. It's, you know, any technology company, it's any life science company has all these parts and pieces. This is not unique to Illumina, it's unique to technology companies. So I love hearing you talk about it. So before we move on from the science to the programs, I just wanted to check in with Marissa and see if we had any questions on any of the things that Jason had covered.
We do not, but if you would like to ask a question, please feel free to type it in the chat box.
Okay.
And I mentioned it, I was in tech support, so I'm happy to answer literally every question. It was my job for 3.5 years.
And I think there's also a little bit of a misconception. When you hear tech support, you know, oftentimes you're thinking about a computer or a platform. And for, you know, Jason, you can expand on this, but, you know, they're working with our customers to figure out how can they use these machines and these sequencers in the best way and in the most efficient way, so that they can be able to do their projects and experiments better and faster. So it's not, when you hear tech support, they are some of our most well-rounded employees who are the most knowledgeable about technology around genomics. So
exactly. You might have students who are interested in science but hate working at the lab bench. It happens. I'm an example of it, but we like talking and chatting. And so tech support, we again have a lot of PhDs, master's students, people who've done grad work in DNA sequencing, who come and their job is to just support customers and teach them how to run their instruments. We'd sit there and chat for hours and help them design experiments. It was a really exciting job and something that anybody who has the mind and interest in science can do, but probably wouldn't consider in high school. So you might open these ideas to some of your students.
Yeah, and it's also very collaborative. It's very, like, I feel like the tech support team, they're always talking to one another. They're working together to be able to solve these problems. And so, again, I know we're focusing on the tech support, but really the idea is there are these, and I'll get to this in a little bit, but there are these really massive misconceptions around what jobs are available for employees. And what you like, think about what you like to do, and there is a place for you at a life science company.
We have a couple questions here. So, the first question is, has Illumina sequencing gotten cheaper because the supplies are cheaper, the tech is better, or a combination of both? What's been the major driver of the decrease in cost?
Yeah, absolutely. So technology has gotten better over the past 20 years. One of the biggest components on any of our instruments is computing power, and computers have gotten better so we can process more data at once, and that just leads to more output. we're also our instruments, the bigger the instrument gets, essentially, the more data output it gets, which means that the more genomes that we can run on it. And so we've made our own technology better and bigger so that it can run that. And that's really because the computing power is there to allow us to do it. And then, I mean, I've never been on the manufacturing side in terms of like, okay, how much does it actually cost for this each individual component as we combine them and whatnot. But I would imagine that as we got the technology, that the just the overall cost of materials to build our products has gone down as well, because we can make them at a higher scale, or the computer chips are cheaper, those sorts of things.
Awesome. Thank you for that. Someone just wanted to make a comment that they started out in tech support too in the late 90s for a competitor of Illumina, but said it was super fun.
Perfect. It's a job that's out there, and there's plenty of us, and I feel like we all had hopefully a good experience, and that we it's a lot of fun. I enjoyed it.
Awesome. And then lastly, someone wanted to know if these slides would be available in a document, in the document section, because they'd love to share this with their students.
Yes, we can make these available.
Okay, awesome.
Right.
And if you're sharing this with your students, there's one thing that I forgot to say. If you're looking at jobs that are unique to a genetic sequencing company, you can take a look at the supporting customers, because there's a job on there called the genetic counselor. And that is really a job that's only found within our space, and something that's really unique. It's really cool. And basically what they do is you get a report of the mutations that you might have in your DNA, but no one's really there to say, this is what you should be concerned about. And genetic counselors work with you, read through those mutations and what might your own DNA might tell you about you, and say, you know, you should be concerned about this type of cancer moving on, or, you know, all these mutations that you have, they're not that big of a deal. I don't think there might be a big concern for these types of things. And so they can help you, lead you through that journey as well, which is a really incredible career and something that's really taking off at the moment.
Yeah. And it is something that's on the clinical side. So if you know, on the oncology side, if you have your genome, if you have your genome sequenced, if they're looking at those different treatments that Jason was talking about earlier, they can help guide you through that as a patient. But then also the genetic counselors on the industry side are helping to create these tests to make sure that, you know, the outcomes are appropriate. So with that, I'm going to move into some of our partnerships that we have, and hopefully, and again, feel free to pop in questions, Marissa, if they're topically relevant for that slide, or we can get to them at the end. So again, Jason, thank you for just making the science understandable, palatable, and really being able to apply the sciences. So, you know, I wanted to look at the different ways that you can take this science and these questions that you might have, but how do you take them into your classroom? And so what we heard earlier was, you know, from a cost perspective. I know a lot of you want to do those hands-on activities, but obviously living in a virtual world right now. And also just from a time perspective, you can't always do the hands-on activities. And so we've partnered with Discovery Education to be able to create curriculum that is applicable in your classroom. And so it's going to cover all types of different topics. So we have genes or the gym to talk about, you know, whether athleticism is genetic or whether it is earned. We have precision medicine, so talking about how a hospital can go from your typical diagnoses and things like that to a precision medicine. So being able to look at those whole genome sequencing, but how does a hospital actually do that? Another one is disease detective, so talking about epidemiology. And so we're able to take these different topics and be able to use the different curriculum. And I'm going to dive into the two types of curriculum that we have. There's classroom activities, and there's what are called digital lesson bundles. And then I'll show you what both of those look like. So if we go to, I'll go to the next slide, and it's going to scroll through what the classroom activity looks like. So all of this I want to mention is open sourced. You'll see the link for this, and it'll be provided. There's a one-pager in the handouts for all of you to be able to download. And all of the classroom activities can fit into a 45 to 60-minute class period, which really gives you that flexibility. So, and they are all, again, they're aligned with next generation science standards, the cross-cutting concepts. So you can see where they actually, it does go quickly, but you know, I really just wanted you to be able to see the depth and breadth around what is in each of these classroom activities. So it's showing all the different careers, it's showing the different handouts that are available, and it's all on dnadecoded.org. So the second one, I should mention that the one that we looked at was called Disease Detectives, and it goes through a disease outbreak simulation to explore the impact of disease mutations. So I'm not sure what could be more topically relevant than that, but you know, something as you're thinking about going into your classroom next year and what you're going to bring into your students, a great option. So the second one is called a digital lesson bundle. And this is a multi-day activity. So it's broken out into 45 to 50-minute blocks of time. And again, they include ready-to-go lesson plans that are aligned with those NGSS standards, Common Core standards, and they have virtual modifications, they have online platforms that they recommend using. And so what you're looking at here is, at the top is the link that you'd click on the website. And then on the right-hand side, it's an educator guide. And that educator guide is, I will say, it is a longer educator guide. They're about 20 pages, but they're very take what you need, leave what you don't need. And then the bottom is the PowerPoint presentations that come with it. So those can be uploaded into Google Slides. You can make the modifications that you want, but you're not having to come up with content from scratch. So each of the slides has speaker's notes, again, that are consistent with the educator guides. So again, there's 14 different topics. So, depending on what you're going through in your classroom, what time of year that you're teaching genomics, and how you can connect it. You can choose whichever one works for you. So each of these digital lesson bundles and classroom activities ties back to careers. And I think Jason, he really talked about a lot of different careers. And so what we have on our Illumina STEM site, which is illumina.com/STEM, are these different career videos. And we have seven different career videos, which you can see, you can see the genetic counselor on there. You can see the clinical lab scientist, a deep learning engineer. What does that mean? And what does that person do? So each of these different individuals that we're showing you who they are both inside and outside of the office. And so each of these slides, this is going to start. Are you able to hear that, Marissa? No, okay. I need to pause sharing and start sharing again. So bear with me 1 second. So with each of these, I'm going to take a minute and show you what the videos look like. And I want to show you for a couple of reasons. One, I want to show you because I think they're really dynamic videos. They show who these employees are inside and outside of the office. And they're not your typical, this is what I do in my day-to-day. And so on the right-hand side of the screen, you can see a career card that goes with each of the videos. And so that's meant to talk about those misconceptions that I mentioned earlier. So what is the job? What does the day-to-day look like? What are some of those outputs and those deliverables that you're going to need to be using and creating in that type of job? And then there's also reflection questions to be able to piece each of these jobs together. So I'm going to take a minute and let this play, and let me know if you can't hear it for any reason.
Hence, you're working. Work can be very intense. You're working on a problem, you're really dedicated to fixing it. I like to run and also have a motorcycle. I got one in college just as a way to get around to classes. Now I just use it for fun and also to clear my head sometimes. I'm Erin Fang and I'm a bioengineer. A bioengineer is someone who applies the principles of engineering to biological processes. Kind of like how a typical engineer would learn about flow through a pipe, a bioengineer would take that knowledge and apply that to flow of blood through a blood vessel, or maybe even flow through a medical device. So you come in around 8:30, check out my emails, start planning my day, then go into the lab, work on an experiment, go back to my computer, start looking at the data from that experiment, do some analysis with statistical software, maybe a break for lunch, come back and have a meeting with the stakeholders for the project and go over what was found, and then maybe spend the rest of the day finishing up a report for that project. When I was in high school, I found out that I had a genetic disease. I knew that I wanted to do something to help people like that. So that got me to apply as a bioengineer to college, and after I graduated, I found out about this amazing company who actually does things to help people with genetic diseases. Favorite part of my job is working with people. It's really great to be somewhere where you know that you're making a difference. Everyone who works here, they're being able to contribute to these products that are going to impact human health and improve things for generations to come.
All right, so that is Erin. She is one of our rock stars. But again, these are great as a standalone resource. They're great accompanying each of those DNA decoded curriculums. And so they're really able to be a plug and play for you throughout the year. So the next partner that I wanted to share is Skype a Scientist. And Skype a Scientist is an organization that we've partnered with, and they're on a mission to make science accessible and fun through personal connections with scientists. And so they focus on connecting the general public, classrooms with scientific disciplines in those fun, meaningful, engaging ways. And you know, we personally, Illumina has tons of different employees in their database, but they have a matching system. So if you're able to explore your curriculum for next year and find where you can plug scientists in to connect with your classrooms, and whether it's about bioinformatics or you're talking about water systems or you're talking about squid, you can really look at the different curriculum, the different topics that you're going to talk about, and where do you want to bring those professionals in. So that's what we have currently. And then I also wanted to highlight something that we did throughout the month of April. So throughout April, we had a goal of connecting with over 10,000 learners through our genomic literacy programs. And the interest was so incredibly fantastic that we were able to connect with over 40,000 learners. You know, our hope is to connect with all types of classrooms, all types of educators, with that focus on making it available to underserved communities, making it available to teachers all around the country and the world. And so, really incorporating our Illumina employees into that experience was really important to us. And reducing the financial barriers, we talked about that in the beginning. So, and again, we want to be able to elevate our resources to our educational partners. So what did we do in the month of April? We have the programs that I've talked about year round. So that's the DNA decoded curriculum, the career videos, the Skype a Scientist, and then we also have a strawberry DNA extraction protocol that's on our STEM website. Depending on where you are in the teaching process, if this is something that you want to do in the beginning of the year as kind of a warm up, it's something that's easy and that you can be able to bring in your classroom. So throughout April, we had over 250 Illumina families attend our Illumina Bring Your Kids to Work Day. We had over 550 educational nonprofit partners and 150 Illumina employees who volunteered. So we had 3 featured sessions around genomics with Skype a Scientist. We still have one tomorrow. It is for more like an elementary middle school audience, but that is tomorrow. So I've given you all a lot of information. Jason's given you a lot of information, but what are the next steps? So I've come up with 3 simple ways that you guys can all bring genomics into your classroom. So the first is plan on incorporating a DNA decoded lesson plan next year. I know that we're in April, we're in the end of April. You guys are in the home stretch of being able to, you're going into summer. So I'm not suggesting that you do any of these right now. But as you think about next year and how you want to, again, incorporate genomics into your classrooms, plan on figuring out which one that you want to do is. The second is register your class for a Skype a Scientist session about genomics or other topics. And then 3 is put DNA Day on your calendar for next year. And that is, this year it's a Monday, April 25th. It's the same day every year. So we can find ways to be able to engage each of you and then all of your classrooms on April 25th or throughout the month of April. So I'm going to move into Q&A. I know we answered some earlier, but if there's additional questions, Marissa, we can answer those now.
There are. So the first question is, are these resources freely available, and can we remix the materials in a way such as making derivatives of the resources mixed with our own materials?
So yes and yes. Yes, they are free. It is one of the guiding principles of our program that the programs and the content that we put out there is open sourced. It is free. So yes, DNA Decoded, Skype a Scientist, the career videos, those are all free. And then as far as modifying it to be what you want it to be, DNA Decoded, yes, absolutely. Take those slides, put them into Google Slides or whatever platform you use, and you can take out the ones that you don't need, or insert a career video into your existing lesson plans.
Awesome. Next question is there anyone that offers metagenomic sequencing access for classrooms under $50 per sample? Would be great for ecology meets biotech classrooms to community science examine local sites.
That's a good question. I don't know any off the top of my head, but maybe it's something that we look into for next year, see if there's any universities that are partnering with local high schools for sequencing metagenomics. I mean, it sounds like a really cool project and something that students could get involved in and connecting and collecting and analyzing those types of environmental samples.
Awesome. Another question was, how can we sign up for a mailing list to be reminded of these opportunities?
So we will be collecting those email addresses through Biocom if we're able to do that, and then we'll be able to share the opportunities with you.
Awesome. Any other questions? Please feel free to put those in the chat box. Or
if you do want to put your email in the chat box so that Marissa has it from this session, we know it's specifically for this topic. You're welcome to do that as well.
Definitely.
And I will say, so this was our first, when we're talking about DNA Day, this was the first year. And again, we had an incredible response. And so we are going to be doing it next year. So if you are interested in being able to be a part of that, please let us know. And also we will likely be reaching out in February So keep an eye out for that. I know it's a long ways away, but just, I know some things go to spam or they're filtered. And so just throwing that out there. And I will say Jason was incredible in supporting our first inaugural year of The Future is Bright.
I just did a DNA extraction with a class in DC this morning and got my DNA necklace for it. It's a lot of fun. It's great for Illumina employees to visit classrooms, and from my experience in it, a lot of the learners have a really good time as well.
It looks like we're getting some thank yous and some email addresses. If there's any more questions, we're happy to answer them. I know that the next section is beginning, but please feel free to type them in the chat box.
Perfect. And if there are any questions that come up before or after, you're like, oh, I wish I would have asked this question. Again, feel free to reach out to Biocom. Biocom, we work closely with them. I talk to Marissa on a weekly basis. So we can be able to connect and help to support you in your classrooms. We want to know how we can help, what topics you want to talk about with your students. And again, we just want to champion the work that you're all doing. So
Awesome.
Thank you everyone for your time. Oh, go ahead.
We have one more question. Will you be adding more of these resources in the future? Yes. So hopefully with the world opening up, we will have more career videos. And on DNA Decoded, yes, we will be doing some different approaches to content, but there will be new content coming out. Awesome. I think that is it for now. Thanks, everyone.
Thank you, everyone.
Thank you.